Let us now learn the operation of electron transport system in the mitochondria. Electron transport system can also be called as electron transport chain. But before you get down with their mechanism or operation, it's very important to get familiar with the system first. And if you know the system, you can excel at their mechanism. So, firstly, get yourself familiar with how are these systems formed, and then be aware of their location. Of course, it is in the mitochondria, but remember, mitochondria has many compartments, isn't it? So you have to specify the location. And to do that, you have to be familiar with the mitochondria, right? And then get down with the main goal. You have to often ask this question to yourself, why it has to exist and why it has to operate, right? And if you can understand the goal, then you can excel at the mechanism. Let me now take you through the compartments of mitochondrion. This is the outer membrane of mitochondrion. This is inner membrane of the mitochondrion. And there's a space between the membranes, which we call it intermembrane space. And then we have matrix here. Now, first you be familiar with how ETS are formed. What makes the electron transport system, right? So if you get down here, there is protein complex 1, 2, 3, 4. So there are 4 protein complexes, and there are also cytochrome C, which we call it as coenzyme Q, right? There is also cytochrome C and ubiquinone, which is represented by capital Q. So these two are very important component of electron transport system because they help in shuttling electrons from one complex to another complex. So they are mobile. Basically, they are mobile. They can move through the membrane and they can transport electrons from one complex to another complex. Whereas the protein complexes 1, 2, 3, 4, they are fixed, all right? They are immobilized. And to transfer the electrons from one complex to another, we need help from cytochrome C and ubiquinone. So this tells us they are very crucial components of the electron transport system. Now you are familiar how electron transport system are formed, right? How are they formed? They are formed by the protein complexes. And where are they located? They are embedded in the inner membrane of the mitochondria. So now you are familiar with the location. Now the next thing is understand their goal. What is their goal? So if you notice here, these complexes are busy involved in pumping protons from mitochondrial matrix to the intermembrane space. You see, from mitochondrial matrix to the intermembrane space. And why they have to pump protons from matrix to intermembrane space? So that they can maintain high proton concentration in the intermembrane space and low proton concentration in the matrix. And why it is so necessary to create such concentration difference, right? Because remember, proton concentration gradient contains potential energy which will be used by the ATP synthase later to generate ATP, which are necessary to sustain our cellular activities. And it is ATP synthase that has the ability to directly generate ATP. But the sad thing here is, ATP synthase, though they have the ability to generate ATP, someone should be there to energize them. Someone should be there to power them. And who's going to do that? Proton gradient. And who creates proton concentration gradient? of course, the incredible system called electron transport system, right? So now you got the clarity why such system has to exist and operate in our mitochondria. So my dear students, be grateful that we have electron transport system that generates proton concentration gradient and powers our amazing ATP synthase, which ultimately involves work so hard in generating ATP, which helps in sustaining our cellular activities. Let me now take you through the operation of electron transport system in the mitochondria. But before that, get yourself familiar with these compartments and components. Take these as inner membrane of the mitochondria, which has incredible electron transport system. Take this as complex 1, this as 2, 3, and 4. Take this as ubiquinone and this as cytochrome C. And this compartment represents intermemory space and this as matrix. Alright, now let's get down with the operation. So NADH, which has been generated by the Krebs cycle, they go on to donate their electrons to protein complex 1. So let's see. You see? So it will associate with protein complex 1 and it will donate its electrons to protein complex 1. And in the meantime, NAD plus is regenerated and H plus is released, right? And as the protein complex 1 gets the electron, they get supercharged, all right? They get energized. Remember, electrons, they contain so much energy in them. So therefore, as complex 1 gets the electron, they get energized. And now they are ready to use this energy to pump protons from matrix into the intermembrane space. So you see, now the protons has been pumped from matrix into the intermembrane space. And then electrons, after their job gets done, their job is to pump protons from matrix to intermembrane space, isn't it? So after their job gets completed, electrons are given off to ubiquinone. So ubiquinone, being mobile in nature, they take away the electrons, all right? And now, in the meantime, from root 2, FADH2, it associates with the protein complex 2, all right? It will associate with the protein complex 2 and then it will donate its electrons, you see? Now the electron is taken by the complex 2 and then from complex 2, it is taken by ubiquinone again. So you see? But the unique thing about protein complex 2 is it will not be able to pump protons because it doesn't get super energized. So sad. Anyway, so electrons are taken by the ubiquinone and it is transported to complex 3. And as the complex 3 gets the electrons, in a similar fashion, they get energized and they uses this energy to pump protons from mitochondrial matrix to the intermembrane space. And now you can clearly see the protons accumulating in the intermembrane space, right? And here the protons are decreasing. And after the complex 3 gets their job done, electrons are taken by ubiquinone. Let us wait. So you see, it is taken by the ubiquinone and then it is transferred to complex 4. And again, in a similar fashion, as soon as the complex 4 gets the electrons, they get energized, all right? they get energized, they get supercharged, and they use this energy to pump protons from matrix to intermembrane space. So by now, at this point, high proton concentration is created in the intermembrane space and low concentration of protons are maintained in the mitochondrial matrix. So you see, with the pumping of protons by the electron transport system, they have created high proton concentration in the intermembrane space and low proton concentration in the matrix, right? So what do we call this? Proton concentration gradient, or you can also call it as electrochemical gradient. And also try to relate with the pH, which is very important. So low proton concentration refers to high pH, and high proton concentration indicates low pH. Remember, pH stands for power of hydrogen ion concentration. So if there are many hydrogen ions, it indicates that it is highly acidic, right? Low pH means acidic. And if there are very low concentration of protons, it indicates that the pH is alkaline. So therefore we say high pH, right? Now you see, this is how proton concentration gradient is established by the electron transport system. Now where do you think this electron will go? And you see, you have to remove these electrons from your complex 4. Otherwise, you're going to suffer. If electrons gets accumulated in the complex 4, it is for sure that your electron transport system will collapse. And you know what will happen if it collapses, right? You know the potential consequences. If electron transport system collapses, ATP synthase will not work and you will not be able to generate ATP, right? Your mitochondria will not be able to generate ATP. And in absence of ATP, our cells do not function. Our cells gonna die and the ultimate result will be death. And we don't want this to happen. As far as possible, we have to remove this electron. So tell me what you're gonna do. How are you going to remove these electrons? It's very simple. Just breathe in. Just breathe in. Hello. So you see, when you breathe in, your oxygen will take away the electrons by preventing the leakage of electrons. You see, electrons are very dangerous species, by the way. If they get leaked out, they'll react with other cellular components. They'll react with other chemical compounds and they will lead to the formation of toxic substances which will not allow our cell to function properly, and it will definitely result in cell death. So therefore it is very important to remove these electrons, and to remove, you breathe in oxygen. They will act as a final electron acceptor. They will take away the electrons and then they will convert it into water by combining with protons which are there in the matrix. And this also helps in reducing the proton in the matrix and helps or contributes in the creation of proton gradient. You see? So that is the significance of oxygen. It acts as a final electron acceptor and it ensures that our electron transport system functions efficiently, and when they do that, we can generate a lot of ATP to sustain our cellular activities, right? And this particular process is known as terminal oxidation. And this is how electron transport system operate. So you see, you understand the purpose first. What was their purpose? To create proton gradient. And why they have to create proton gradient? So that ATP synthase gets powered and generate ATP. And to pump protons, what do they need? they require energy, right? So from where do they obtain energy? From the electrons. And what are the sources of electrons? NADH and FADH. And from where they get FADH and NADH? From the Krebs cycle, isn't it? So you see, that's how steps in cellular respiration are interconnected. One cannot operate without the other, and it operates in a systematic manner. They cannot operate randomly. They have to operate in a systematic manner. Now, in the next lesson, we will learn how ATP synthase functions and generates ATP.